Okay, so thank you. So my name is Ben Cumming, and I'm going to be presenting another tool, a simulation tool similar to NEST that we saw in the last presentation. So this is a library and a Python interface for simulating networks of neurons. It operates at a different sort of spatial scale than the NEST. So in NEST we saw focuses very much on the network dynamics and uses simplified models of cells, which allows you to scale up and model much larger networks. Arbor is a tool that's designed to give people the ability to have much more detailed models of cells. So a cell isn't treated as a single point with some simple state attached to it. It can be have a complicated morphology, and we can model things like ion channels and more complicated synapse dynamics. So this Arbor is something that has got a much more recent or briefer history than other established tools. So it's been developed entirely within the HBP ever since a meeting when I first visited here in Jülich about 4 years ago. So I visited about 4 years ago, and a little team has grown up around that. And a list of basically all the people who have contributed all within the HBP. So we've got from Jülich, we've got Arne, who's here today, and Alexander, who would like to be here, but I think he had an emergency to see to. And also we've had Susan, who gave the presentation this morning, has also made a small contribution. And then we've got a team from the Swiss National Supercomputing Center, where I'm based, that have been contributing. So in this talk today, I'm not going to go into too much detail about exactly what Arbor models. We're not going to give a big overview of our multi-compartment cells and this sort of thing, because it's a big topic. And I hope that there are some people here in the audience today who are interested in this topic. And so this is really targeted at those people. So I'm going to give a brief introduction to Arbor and why we tried to develop a new simulation library. Some basic building blocks of Arbor models, what you need to know and do to make a simple first model in Arbor, and then the Arbor workflow, the process that one would go through to build a model. And then once I've finished, we're going to have Anna's going to take over and do a little hands-on exercise with the Jupyter Hub notebook, where you'll get to actually get a ring model up and running. And then at the end, I'll just do a little demonstration out the front where I'll show Arbor scaling using MPI and multi-core processors, and show how it can be used to run simulations efficiently in parallel. So before we start, I've got just a couple of questions just so I can understand the audience. Is anybody in the audience interested in or going to work with or currently working with multi-compartment models? So we've got a couple. I think we've got maybe only 2 or 3 people. So, and has anybody here ever used NEURON, or do they use NEURON? Okay, we've got one hand goes up over there. So if yes, then this talk might be interesting. I hope the rest of you will also be able to get something out of this. So Arbor is available on GitHub as open source software, just like our NEST we saw before. So you can go to our GitHub page, and the software is there, along with instructions on how to install it and how to run some examples and tests. We also have our documentation, again, like NEST. It's at readthedocs.io, and this documentation is quite recent. We've been working mostly on the C++, low-level backend, and only recently, the last 6 months, we've really started putting effort into the Python wrapper. And the Python wrapper, as we're going to find out, is the main way that users will interact with the tool. So we've been making documentation for that, but we're not going to pretend that it's complete. So we want to encourage people to have a look at the documentation as they go along and give us any feedback they have about what we could improve or what you found confusing. So I'm just going to start with a little overview of what the challenge is that we're trying to meet. And when people try to build models of using multi-compartment cells, so complicated cell morphologies, these models that they build are complicated on multiple different levels. So every group is researching some different part of the brain, some different hypothesis, and they've got they have completely different models that they want to run, and different parts of the brain have got, you know, are very diverse and very different. So people want to have complicated cell morphologies. They want to have different types of cells, and then they want to be able to have ways of describing how their ion channels and the synapses and all these things are placed and distributed on a cell. And some people might do it using rules. So they'll say, on all the dendrites, put the following things, and maybe have some random variation in there. Others might say, well, no, we're actually going to take all the cell descriptions from measurements we've made in the lab from specific cells, and we're going to have just a very explicit description of what's on the cell. So already you've got quite a lot of complexity. You've got to handle quite a few different sorts of workflows. People have different ways they want to do things. And then we come to how do we connect the cells together. Some people have simple hypothesis or rules-based things saying, well, we're going to have this population of cells here and this population of cells there, and we're going to have this many incoming, you know, connections from this population to this population, and it's going to be randomly drawn from some random distribution. So you write down a simple rule, and then you generate the connections. Whereas other groups actually go and fill up a three-dimensional volume with a bunch of these cells, and then they look at where they intersect, and they build up connection maps that way. So again, they're coming with an explicit description of exactly how things are going to be connected. And then finally, people want to measure and store different things. Some people might be quite happy with just the spike times that come out. Other groups will actually want to have voltage traces from individual locations, or maybe from every location in a cell. Others will want to get currents and gating variable state. So you have to be able, any sort of modeling approach or tool that you give people has got to give them the ability to describe this diverse different, you know, workflows and diverse different models, and then describe how they want to, you know, what they want to measure, what they want to observe. Here's just an example of, you know, the sorts of complexity you can have. This is a simple Purkinje cell, a single Purkinje cell model, and one group might say, oh, well, we actually want to, you know, simulate this whole thing in all of its intricate detail. Others might come up with much more reduced and simpler models. And you've got to be able to handle this sort of complexity, because this looks completely different from another type of cell, and people want the ability to describe all the cells and all the things. And they get upset when they can't. The other challenge we have is performance, because people want their simulations to run fast. And often to do new science or to do something interesting that hasn't been done before, you want to run larger models, or you want to run more simulation runs, or you want to simulate for longer. Instead of simulating 1 minute of activity, you say, oh, I want to simulate an hour or a day of activity. And the problem is that we have a finite amount of resources. You know, we've only got one computer or two computers, or we've only got this many hours on the JSC supercomputer to do all of our simulation in. So we really need to use those resources efficiently. So it's got to be fast and with low memory overheads. And the big problem here is that if you want to do efficient simulation, then you really need to organize all of your data and your information in memory very carefully, and then have the algorithms access that information and process it in a very structured way. But this is kind of at odds with a general modeling approach that lets people build arbitrary and complicated models. How do you map all of those models onto efficient data structures? And it's possible to write for a given model, to sit down and spend, you know, 6 months handwriting code in C and CUDA or whatever your, you know, chosen language is, that will run that specific model very efficiently and get good results. And every year we see, you know, a handful of papers coming out showing wonderful results on GPUs or different architectures. But it's very, very difficult to write a generic framework where people can put in, you know, these generic models and have it run all of those models, or the majority of those models, very, very efficiently. And that's why there's only really two tools that are widely used in this sort of area. There's NEST and NEURON, because they've managed to do a reasonably good job of they've given people the ability to do very generic modeling, to model all the things, and they do this quite efficiently. So NEURON is another program simulation library that's very similar to Arbor, and its great success has been the flexibility it's given users, and that it runs reasonably efficiently on multi-core CPUs. But now we've got GPUs, and we're getting more and more cores and more vectorization and more of these things you heard about yesterday about different, you know, sorts of architectures coming along. And if we're going to be able to keep giving people performance improvements, we need software to run on those different architectures. And Arbor is designed to, from the ground up, to basically not just run well on multi-core CPUs, but to run efficiently across a large range of architectures. So, so specifically, it's a library for simulating large networks of morphologically detailed, so detailed cells, we are spiking neurons. And this is a key point. It's usually for large networks because that's where we start to really care about performance. When we want to run models with hundreds of thousands or millions of cells, or we want to run 100,000 models with one cell in each model because we're looking for some parameters. And it runs across the diverse different hardware that's available, particularly to HPP, any sort of hardware that's currently available to HPP researchers through the HPC infrastructure. It runs efficiently on that. So it supports GPUs, it supports vectorized multicore. And you learned about vectorization and multicore yesterday in the talk about, I forget what the title of the talk was, but where you learned about number and tools like this. And it also runs across clusters, which means that it will run across multiple nodes in a distributed fashion. And it's got two interfaces, a low-level C++ API. So C++ is a low-level programming language that the library itself is designed in, but that's not too useful for general users because it requires a lot of specialist knowledge. So we also have a user-friendly Python interface, which is what we're going to be looking at today. So now I'm just going to go through a few basic fundamental building blocks, and this is very much a practical how-to, just an introduction to the bits you're going to need to build models. And the things that we need to understand are cells, connections, spikes, and events. So this is a similar list of basic ideas that we needed to understand to learn about NEST earlier today. So the first building block we have are cells, and Arbor supports multiple different cell types. So we've got support for leaky, integrate, and fire, LIF, so single compartment cells, and spike source cells, which are just cells that you can use to generate sequences of spikes to inject into the network. But the most important in the context of Arbor is what we call cable cells, which are multi-compartment cells. So you saw the picture earlier of that cell, and it had like a soma, and then it had this branching cable-like structure. And the way we model these cells is we break them down into basically a set of connected branches or connected cable sections. So we have these one-dimensional line segments that branch, and we model a cell as a bunch of these connected line segments. So if we in Python, and we'll do this practically later on, if you've set up your environment correctly, you can just import Arbor and then start using the features of Arbor. So to make a simple lift cell, we would call the arbor.liftCell function, and then we can modify the lift cell's parameters. So here we've modified the parameter VM, and you can type in help, or you can print this out, and you'll get a list of all the available parameters and what you can do to modify them. And this here, we've got a simple little function that helps us to make a cable, what we call this cable cell with a random morphology, because sometimes it's nice just to be able to generate a random cell with some different properties on it and use that to build up a demonstration network. So we're going to use this feature here today to build that network because we don't want to go through the hassle of actually constructing, a full 3D cell reconstruction. So I'm going to skip over this because most of the audience isn't particularly interested in cable cells. But there's a little bit of theory. This basically describes how we model. We break down the cell into a set of these one-dimensional cable segments, and then we model the electrical conductance and the potential by using something called the cable equation, which is a partial differential equation. And all of this is, you basically describe the cell and say run the simulation. The user isn't really involved in this solution step here. But people who are interested in this, we use a very similar approach to what NEURON uses. And a key point here is that once you've built the cell morphology, you then need to describe the synapses and where they are, and the properties of the synapses, and then the ion channels that are going to, dictate the dynamics of the cell. And we use something called NModel, which is a simple little language that was designed for the NEURON simulation package. And NModel is a very simple little language that lets people describe the dynamics, describe how an ion channel works. So we also support those descriptions. The next building block is what we call cell members, because we often, well, particularly with multi-compartment cells, we really need to be able to refer to things that are on cells. Because on multi-compartment models, we can have many synapses on a cell, we can have many spike sources, we can have many different things. We need to be able to say, I want the 5th synapse on cell number 4, for example. You need to be able to refer to things. And we refer to these things as cell members. So every single cell in the model has a unique number associated with it. And so if we've got n cells in the model, the cells are all numbered contiguously from 0 up to n cells minus 1. So we always use 0-based indexing. So we start counting at 0 everywhere in Arbor, which is the usual way that Python works, and C++ as well. So first of all, we use this GID to identify which cell we're talking about, and then we use a second number, the index, to refer to the thing on that cell that we're referring to. So cell member always has this structure. It always has a global identifier that it identifies the cell, and then another that identifies which thing on that cell we're talking about. So we could create a cell member 42.0, which refers to the first thing on cell 42. Now, which thing it's referring to depends on the context. If we're talking about synapses, then this is referring to the first synapse. If we're talking about spike detectors, we'll hear about them later on, we're talking about the first spike detector. If we're talking about gap junctions, the same thing. So at the moment, this is just an index. And then what it actually refers to will depend on the context in which you use it. The next thing we have to do, once we've got a model with a bunch of cells, we need to connect them together. And cells are always, and you saw this is a very similar process that we saw earlier with NEST, that we had to describe how to connect two cells together. In Arbor, we use what we call a connection, and every connection has a source. That is where it has a source, which is usually the source or where a spike will originate. And that will be a cell member. So that's going to be saying the source for this connection is this specific cell and this specific spike detector on that cell. It has a destination, which is a synapse on another cell where that's going to, when a spike is created, there's going to be an event delivered to that spike, to that synapse. And again, that's a cell member. And then it has a delay, which is, when a spike occurs, there's a delay before an event or the response arrives at the synapse on the postsynaptic end of the connection. And this delay is, you describe this delay in milliseconds. And there is also a wait. So when the event is delivered, it's delivered with a wait or a value attached to it. So you can have multiple connections coming into a single synapse, and then each time an event comes along, it will have the wait associated with that connection, which can be used. And this wait, how this wait is used really depends on the type of synapse and how that synapse interprets that. So there's no units associated with this wait. It's specific to the connection type. So again, if we wanted to make a connection that goes from cell 42, the spike detector on cell 42, to the 104th synapse on cell number 2, we would make two cell members for the source and the destination, and we would then create our connection like this, source destination, and specify the delay and the wait. So there's a lot of detail on this slide, but we basically, we have an important distinction to make is the difference between what we call spikes and events. A spike occurs usually when you'll have a detector usually on the soma of a cell, and when the voltage in that compartment in the soma crosses some threshold, this is considered to be a spike. This is a spike event. So a spike will be generated when this threshold crosses, and then we have events. Events are what's delivered to the synapse. So when a spike source is connected to a synapse on another cell, at some point in the future, according to the delay time of the connection, there's going to be an event that is delivered to that synapse to say, hey, a spike occurred on the other end of this connection. So we call the things that are delivered to the synapse an event, and we call the original thing that created this the spike. So it's important to note that you can have one spike, but then you can have many, many connections coming out from that spike going to other synapses. So one spike will generate many events. And unfortunately, different simulators all have different, words that they use for these things, so it can all be a bit confusing for new users. Okay, we've got things called event generators, and I think we'll see those in the hands-on. I won't spend too much time going into those now. We'll just check how I'm going for time. So these are the basic building blocks for that you have to understand to start building a model. Because once you can make some cells and connect the cells together, then you can start, and then just maybe put some events into the system to start the system off. Then you can start to build some simple models. The next thing we have to look at is the workflow. How do we put these things together to actually build a model and run it? And the first starting point for Arbor workflows is defining the model using what we call a recipe. So this is a little bit different from the approach that you saw with NEST and that you've seen that you would see with NEURON, where you would, in NEST and in NEURON, you basically just start to say, you start typing in, I've got a population of cells, or I've got a cell, and I've got a connection between cells, and you explicitly enter in the model and then say, run the model. In Arbor, we separate the description of the model from the actual running of the model. We come up with something called a recipe. So recipe is just a description of the model. It's not actually the model that we're going to run, and that'll make sense in a bit, hopefully. And what you do is, when you want to describe a model in Arbor, you sit down and there's a common interface that you use for describing the different components. And it uses a cell-centric description. So you basically have to write a function that said, for a given, when someone gives that function a global identifier, so the unique index of a cell, you should be able to return what type of cell it is. Is it a cable cell, a leaky integrating fire cell, etc.? A description of the cell. So if it's a cable cell, then the description would be the actual morphology and the ion channels and the synapses on that cell. And then the incoming connection to that cell's synapses. So a list of all the connections that come into that cell. So you basically write you write this interface that will describe all these things. And there's also the gap junctions on a cell, which we're not going to look at today. And you also have to say how many sources, targets, and gap junction sites there are. We'll see this in the hands-on. So here's an example of how you actually write a recipe, because recipes have to conform to this well-defined interface. So the way we do this is we use inheritance from a class. So you probably haven't seen this, but you can define interfaces. You can define an interface in Python using a class. And this interface says everything that implements this has to, for example, have a function called numCells that will return an integer. And this function numCells is basically the number of cells that are in our model. And it also has to have a function called numTargets that takes one argument, which is the global identifier, the GID, and returns how many targets are on that cell. And so we always follow this pattern here. You define your recipe, and this, in my example here, I'm defining a recipe that's my 10-cell model. And it inherits from arbor.recipe, then it implements a list of functions. This is only some of the functions that have to be implemented. There's a longer list of them, and we'll see those in the practical again. But this recipe defines, so every single cell kind here is always going to be a cellkind.cable. Because you can see this function here, it takes a GID, but it ignores the GID. It just says, every single time you ask me, I'm just going to say that it's a cellkind.cable, which means that this is a model that's comprised entirely of cable cells. And then someone might ask for the connections on the cell GID. And we're going to return, this here returns a connection that has, for the source, it has the first spike detector on the first cell. So every single cell is basically going to be, and then for the target, the target is the first synapse on the cell we're talking about. So this is going to be a model where you'll have the first cell in the model that's connected to every other cell. So it's a one-to-many model. It's not a very practical example in terms of our reality, but it's a good little way just to demonstrate how you would do this. So we're returning a list here with just one connection. But in a typical model, you would actually have, hundreds or thousands of connections, up to about, the order of 10,000 connections coming in. So this would be a very long list of connections. And here you can see that we've got a lot of flexibility in how we define these. You could read these off a file on the disk and return the list like that, or you could have some rule that you could randomly generate them according to some rule and return them. So, and then you can create one of your 10-cell models, for example, and then you can start to use this interface. I'm going to skip over this because it's the idea. Well, yeah, I'm running a little bit low on time. But if you want to understand a little bit more about performance, because Arbor is designed for performance, and these are basically some tips for maximizing the performance. The next thing we had to do is we have to define the hardware we're going to run this on. So when we defined that class, we didn't actually create a model. We just defined an interface that something else will be able to use to build the model. So that's the model. And the next thing we need to do is describe where we're going to run the model. And we do this with what we call a hardware context. Because when we run a model, we might be running it on a laptop, like on my laptop here, which has got 2 cores. So I'm just going to be using 2 threads, or 2 cores to do the computation. I might be running it on my desktop PC at work, which has got 4 cores and a GPU. So I might want to say, I want to run this model on 3 of those cores and using the GPU. Or I might be running it on the UQueen system here in the compute room, which has got multiple nodes. So I might want to say, on each node, I want to run it using 20 threads, and then I want to run it distributed. So distribute the whole model over however many nodes we've got, let's say 100 nodes. So we have a separate way of describing exactly what hardware we're going to use to run this. And we do this using what we call a context. So every single context always has at least a thread pool. So a thread pool is the number of CPU threads or cores that we're going to use to execute the model. And then optionally, you can also give it GPUs to use or a distributed MPI context. So you might remember yesterday from you learned a little bit about MPI, which is what we use when we have multiple nodes and we want to get them to communicate together. So we have an MPI interface that you can optionally use. So to describe the hardware resources, we use an Arbor context. So if you just create one by opening and closing the brackets without providing any arguments, that gives you the default context, which is just 1 CPU thread. I think we're going to change that pretty soon to actually try and automatically detect how many cores are on the system it's running on and use that number. But at the moment, it just uses 1 thread. Then if you want to do something a bit different, you can start to pass in arguments. So you can say, I want to use 12 threads. And on the Eureka, the system, yeah, these names are too confusing. They all start with JU. On the Eureka system that we're going to be using today, each socket has got 12 cores. So on that system, I would say I want to use 12 cores, and I can explicitly, if I want to, I can say I'm not using a GPU. Or I can say I'm using GPU 0, because whenever you have GPUs available, they're always, let's say you've got 4 GPUs, they're always going to be numbered 0, 1, 2, 3. So you have to specifically say which GPU you want. And we're going to add a function which basically you can tell it, just go get me whatever the best GPU is that's available. But you explicitly say that. And then optionally, you can also provide an MPI communicator. So you were introduced to MPI for Pi yesterday, and you saw this MPI.com world, which is the default communicator. So if you want to use MPI to distribute the problem, all you have to do is say MPI equals and then pass in the communicator. We have other interfaces for doing this, but this I think is the simplest one. And here's like a really complicated one that you might use if you're on Pitstein. That's our big system at CSCS that has 1 GPU on every node. You would say, I want to use that GPU, and I want to use 8 threads, and I want you to distribute this work over the network. So now we've got a description of the model, and we've got the hardware resources we want to run it on. The next thing we have to do is to then distribute this model over those resources. We have to figure out which cells go onto which MPI ranks, which cells go onto which GPU. The work has to be distributed. And we do that using what we call a process called domain decomposition. And we have an algorithm called the partition load balance, which just does this automatically for people. So once you've created your context, here I'm creating a context that has 4 threads, no GPUs, and it uses MPI. And I've created a recipe. So somewhere else, I've created my recipe, and I've called it my recipe because I'm not very original. So I've got my context, which is the hardware. I've got my model description. And now I ask this algorithm here, I pass them into this algorithm, and it returns a decomposition. And then if you want to, you can do a print on that decomposition and start to explore it. And it will basically say exactly which cells are going to be run on which hardware resources. So now we've got all the basic things we need to build a model. We've got the recipe, the context, the hardware context, and now we know how to distribute that recipe or that model over the resources. The final step is to build a simulation. So a simulation is where we've actually taken the model and distributed it and built all of the state, all the things we need to do to actually run the model. Because if we're running the model on the GPU, the state or the things that we have to build look very different from if we're running it on a multi-core CPU. So we have to build this simulation state, and we do that using an Arbor simulation. So we pass in the recipe, the decomposition, and the context, and then we have a simulation. And then we can run the simulation. So immediately we can say run the simulation for 1,000 milliseconds, so 1 second, and we provided time steps. This is our time step size. And I think this is the default time step size, so you don't actually have to supply that. And here you can see this is a very nice feature. We can decide at runtime, depending on how Arbor was configured, we can decide at runtime whether we want to use MPI or not. We don't have to recompile Arbor or use a different version of Arbor depending on whether we want to use MPI or not. You still get the choice about exactly how you want to run it from a single installation of Arbor. So does anybody have any questions about this so far? Checking the time. Now, the final thing that we want to do is we want to actually get some results out of this, because when we just created the simulation and ran it before, we're not actually measuring anything. So we have to actually say, before we start running the simulation, what we would like to measure. Now, at the moment, the C++ backend supports users being able to print out, record voltages and currents and ion states and all these sorts of things. At the moment, we've only implemented the measurement of spikes in the Python interface, and it's just a matter of time before we get around to wrapping the rest. But let's say we wanted to record the spikes that are generated by the model. The first thing we would do is we would create the simulation, and then before we run the simulation, we attach a spike recorder. So we keep a hold of this recorder, we run the simulation, and then once you've run the simulation, you can then do what you want to with these spikes. You can save them to file, or in this case, we're just looping through the spikes and printing them to the screen. And for this particular model here, this is an example of the sort of output you might get, saying that we had a spike on cell 0 at this time, and then cell 1 had a spike at this time. And this is you're going to become very familiar with this model, because this is the one that we use in the demonstration. This is what we call a ring model. So you have a set of cells, and they're basically connected one in a ring like this. So one connection per cell to the next cell in the ring. And if you set things up correctly, it will basically, a spike will be generated on one cell, which will send an event to the next cell, which will generate a spike, and they go round in a ring. So you end up with a sequence like this, 0, 1, 2, 3, 4, 5, 6, 7, 8, 9. And then if you get to the end of the ring, it will loop back to the beginning again. So this is the workflow. It's what I dubbed here the Arbor 5-step method. The first thing is you create a recipe, which is a definition of what we're going to simulate. Then a hardware context, which is definition of where we're going to simulate it, what hardware we're going to simulate it on. Domain decomposition, which is basically a map from the recipe to the hardware. Then we actually create the simulation, which is the everything ready to go and ready to run. and then we execute the simulation. Now, one thing you might be saying is, that's a lot of steps. Yeah, that's 5 separate steps I've got to do. When I looked at Nest earlier, just sat down, made some cells, connected them, and hit run, and my model ran automatically. Why do we have to go through all of these different steps? And it's a design decision that was made because we started building Arbor specifically with the idea of running it on all these different computer architectures. So we said right from the beginning, this is going to have to run on MPI, and it's going to have to run on GPUs. People need a way to describe this. And we decided we've made all of these steps very, very clear and separate. So it's actually much easier to understand, because for a very small model, it's very easy just to say, make a few cells, connect them, and run the simulation. But as your models get larger and larger and more complicated, after you've been working on them for a week, they're usually, hundreds or thousands of lines of Python code. And at some point, they start to become very difficult to understand. The Arbor approach, we've tried to really split it into different components so that things are quite well defined. If you want to say how many cells are in the model, there's a very well-defined spot to go and do that. If you want to say what does the 50-second cell in the model look like, there's a well-defined interface for doing that. Whereas if everything's been built in an ad hoc manner, this sort of information is hidden in a Python script somewhere and becomes very difficult to find. So, and we do this by having a clear separation between the model description, the hardware, and the simulation state. So people who are coming from using NEURON will notice that these things are now completely separated. They might then say, well, there's quite a lot of steps I've got to do, creating context, partitioning domain, etc. But what you'll find is that when you try to use MPI with NEURON, it's a completely different workflow, and the user actually has to kind of manually do this process, has to manually say which MPI rank am I on, and then manually create the cells. And they actually have to do this process kind of implicitly, whereas Arbor does all of that for you and lets you have a much clearer description. So this is a case, again, of once a cell starts getting more complicated, this sort of structured approach makes more sense. And Arbor's approach finally encourages some good practices. It encourages people to make reproducible models. One problem we have is that if you run a model 2 times, one after the other, you would like to get the same answer. If you run a model using 10 MPI ranks, and then you run it again using 100 MPI ranks, you would like to get the same answer. But it turns out that this is usually not the case for models that have been built in an ad hoc manner, because typically somewhere in a model, for example, you're going to use random number generators. You're going to use a random number generator, say, to build some random connections. So you can say every cell is going to have 1,000 random connections drawn from this other population of cells. So you'll use a random number generator. And random number generators behave, unless you're very, very careful, because when we use a random number generator, we usually seed it. We give it an initial seed value, and then it will make a reproducible sequence of random numbers. But if we seed them differently, or we loop over cells in a different order, we will get different random numbers used to generate each cell. Arbor breaks everything down so that you're always using the global identifier of a cell as the thing that you start with when you define a cell definition. So that makes it much easier to say, okay, when I'm making cell with a particular GID, I'm going to seed the random number generator that I use to make the morphology with that GID. I'm going to seed the random number generator that I use to make the connections with some hash based on that GID, and so on and so forth. It provides a much saner interface for people to make reproducible models. And at the end of the day, it's easier to understand. You will find that this approach is easier when you start having larger models, because now when we're looking at the model definition, there's nothing about the hardware mixed in there. There's no confusion between the two, and so on.